The enzyme we chose for our presentation is the organism lactase. Now, the main trait for this enzyme is for metabolizing lactose.
Transcription is the process where the information of a DNA strand is copied onto a new messenger RNA. It occurs in the nucleus, and the main purpose of it is to make pre-mRNA from DNA. mRNA contains a ribose, one of the 4 nitrogen bases, also known as AUGC, and a single-strand structure. It is also important to note the structure of a gene. A gene is made up of 3 sections: a promoter, the RNA coding sequence, and a terminator.
So to begin, transcription has 3 phases: initiation, elongation, and termination. During initiation, an enzyme called RNA polymerase attaches itself to the promoter of a gene, where the double-stranded DNA is cut into 2 pieces through a process called RNA synthesis. One side is called the template strand and the other being called the non-template strand. The template strand is what will be used to transcribe the DNA into pre-mRNA. After the DNA is split, elongation occurs. During elongation, the process of RNA synthesis is still occurring. Ribonucleotides come into the RNA polymerase and begin to be synthesized in the 5' to 3' direction. On the RNA molecule, complementary base pairing occurs, where the complement of the base on the template strand is coded onto the RNA strand. When the RNA polymerase reaches the terminator, the transcription is complete. The polymerase breaks away from the strand and the mRNA molecule floats off to start translation. During mRNA processing, this step is used for the extraction of introns. Introns are segments of DNA or RNA that does not code proteins and interrupts the sequence of genes. To remove the introns, a procedure called RNA splicing occurs. To begin RNA splicing, an assembly of helper proteins is created. These proteins act as a guide for small nuclear riboproteins to form a spliceosome. The spliceosome brings exons on both sides very close together, where one end of the intron is then cut and folded back on itself to join and form a loop. The spliceosome then cuts out the introns, leaving only the exons, which contain the coded information for proteins. After the introns are removed, a cap and tail is added to the RNA segment. The cap is used as a signal for ribosomes to attach to the segment, and the tail is used for the stability of the mRNA segment.
The translation takes place at the ribosome inside of the cytoplasm. We got the ribosome from transcription. The main purpose is to make protein from mRNA. The ribosome structure is made up of 2 subunits composed of rRNA and proteins. The ribosome will read 3 mRNA nucleotides at a time. The 3 mRNA nucleotides are called codons. Codons match for the delivery of a specific amino acid. Amino acids link together to form a single protein, and the start codon is AUG.
Translation of lactase, like all proteins, starts with a ribosome attaching to a start codon with the code AUG on the 5' end of a specific mRNA for lactase made in transcription. The ribosome then attaches a tRNA to the mRNA. This tRNA and its subsequent amino acid determined by each 3-letter code in mRNA. This tRNA and its subsequent amino acid is determined by each 3-letter code in the mRNA. An A in the RNA bonds with a U in a tRNA, and the C in mRNA bonds with a G in a tRNA, and vice versa. The 3-base code in an mRNA is a codon, and the corresponding 3-nucleotide code in a tRNA is called an anticodon. In lactase, the first amino acid brought, aside from methionine, to the start codon is a glycine on the tRNA with an anticodon GGG and a codon CCC. Next, the ribosome moves down the mRNA towards the 3' end. Another tRNA determined by the next codon of the mRNA sequence is attached. The first tRNA dissociates and leaves behind its amino acid that bonds to the amino acid on the next tRNA. This process is repeated, forming a polypeptide. Our protein lactase is an enzyme. Its specific function of lactase is to break down lactose. This gives humans the trait of the ability to digest lactose. It breaks this disaccharide sugar into a glucose and a galactose. The purpose of this is to make it possible for humans to digest lactose and use its energy.
Termination. Stop codons tell the ribosome to stop making protein. Lactase consists of 1,927 amino acids.
Mutations can have a large impact on the lactase enzyme, something that is found in many people's lactose intolerance. This can either be caused since birth by a mutation in the gene LCT, or it can be caused by the decline in production of lactase as a person ages. In the case of the mutation, it is referred as congenital lactase deficiency, also known as CLD. This condition can be caused by many different mutations. However, the most common mutation causing CLD is a nonsensical substitution mutation, referred to as FIN major. The mutation obstructs the production of the polypeptide used to make lactase.